All right, so I'm back again for the anatomy of the ear. The ear is the anatomical structure associated with our sense of hearing. It's divided into three regions. We have the external or outer ear, we have the middle ear, and then we have the inner ear or the internal ear. Each region plays a role in hearing. However, only the inner ear is associated with a sense called your vestibular sense, which is important to our equilibrium, and we'll talk about that a little bit later. Here is a picture of the ear. This is the outer ear. Goes to from the external ear, the auricle or the pinna. There's the earlobe. You have the external auditory canal. The outer ear ends at the tympanic membrane. Some people will refer to the tympanic membrane as the eardrum. The middle ear is made up of the eardrum, the three associated ear bones, and this little area right here. And then the inner ear is going to be the semicircular canals, the vestibule. We have something called the round window, the pharyngotympanic tube, which runs to the pharynx, the tensor tympani muscle, which helps to oscillate the nerves and make our sound. If you've ever had a child or known of a child that had to have eustachian tubes installed, those tubes are installed in this tube right here, so the ear will drain and they will stop getting so many ear infections. The external part of the ear, called the pinna, or more correctly called the auricle, is made up primarily of elastic cartilage, with the exception of the lobule or the lobe, which is primarily adipose tissue. It is covered with skin. The job of the ear is to funnel sound waves into the external auditory canal. The external auditory canal, also known as the external auditory meatus, is a curved tube through the temporal bone, terminates at the tympanic membrane. Again, common name for the tympanic membrane is the eardrum. It is lined with modified sweat glands called ceruminous glands. These glands secrete your earwax called cerumen, which is a waxy yellow-brown to gray substance that lubricates and waterproofs the canal and the tympanic membrane, traps and removes debris before it reaches the tympanic membrane. Lots of differing opinions on earwax and earwax removal. Should you or shouldn't you use Q-tips to clean the earwax out of your ear? I've had students in the past say, is there such a thing as having an ear that is too clean? And the answer to that is absolutely yes. But it's also just likewise, there's also such a thing as having an ear that is too filled with wax. Again, this is the outer ear. There's the pinna, the lobe, again, primarily fatty tissue, external auditory canal. The tympanic membrane is a thin sheet of epithelium and connective tissue, separates the outer and middle ears. It is somewhat cone-shaped with an apex or tip pointing into the middle ear. It's connected to the tiny ear bone or the ossicle. Enables energy of sound waves to reach the inner ear. It vibrates, and that's what starts moving the little bones of the ear, which give us our ability to hear. The middle ear is a hollow, air-filled, mucous membrane-lined chamber within the temporal bone. The membrane forms a boundary laterally and walls off the inner ear, and defines the medial boundary. The auditory tubes, or the pharyngotympanic tube, connects the nasopharynx with the middle ear. Its job is to equal air pressure on both sides of the tympanic membrane. Pressure differences between the chambers reduce the ability of the tympanic membrane to accurately vibrate, which negatively affects hearing. If you've ever been up in an airplane or driven up into the mountains and your ears start to sort of swell and feel like they're going to pop, and you need to, one of the things you can do to equalize that pressure is to hold your nose and like that, and that will equalize the pressure and sometimes get rid of that feeling of having a stuffed up head. The auditory ossicles are the three tiny bones of the middle ear. They are named by their shapes. They're connected to one another at synovial joints. They form a bridge that links the tympanic membrane to the outer wall of the inner ear. You have one called the malleus, which is shaped like a mallet or a hammer, ergo it's named the malleus. It's connected to the apex of the tympanic membrane. The incus is anvil-shaped, which forms the midpoint to the bony bridge. And then the stapes, which is a stirrup-shaped bone, it's connected to the oval membrane in the medial chamber that covers the oval window. The malleus, incus, and stapes are also referred to as the hammer, anvil, and the stirrup. In our lab, we have a nice picture here. I don't know how well you'll be able to see the picture, but I'm going to show it to you. Hang on, let me get it folded correctly so it'll stop bending. There are the three ear bones, the hammer, the anvil, and the stirrup, lying next to a dime. So all three of those bones will fit very nicely on the surface of a dime. So these are very, very small bones. The oval window is a boundary between the air-filled middle ear and the fluid-filled inner ear. These ossicles amplify and convert incoming sound waves in the air into movement in that fluid, which then sends the signals and does the physiology of hearing. You have two tiny muscles attached to the ossicles. The tensor tympani muscle originates from the wall of the pharyngeotympanic tube and inserts on the malleus. It tightens the tympanic membrane by pulling it medially or to the middle. The stapedius muscle originates from the posterior wall of the middle ear and inserts on the stapes. It reduces movement of the ossicles as a unit. So one muscle moves the three ossicles, the other muscle stops the movement. Both muscles are components of a reflex. This prevents damage from loud sounds by reducing the movement of both tympanic membranes and the ossicles. And again, here is the middle ear. There's the tympanic membrane, the hammer, the anvil, and the stirrup, the malleus, the incus, and the stapes. The stapes is sitting in a structure called the oval window. Some inflammation of the middle ear cavity is referred to as otitis media. If you or your children have ever had an ear infection, more than likely this is the one they had. It's one of the most common infections in children, almost always preceded by an upper respiratory infection. So the child has an upper respiratory infection that leads to an inner ear infection or otitis media. Common symptoms, ear pain. If your young baby that is not old enough to talk and tell you yet is constantly pulling at their ear, that is a sign. Fever, impaired hearing, irritability, and in some cases vomiting, depending on the amount of infection. The vomiting is generally going to be caused by the infection has stressed to the point where it is starting to now affect the middle ear, or excuse me, the inner ear. The swelling that is occurring in the middle ear is causing a disruption in the inner ear, and that's where the vomiting comes in. Acute otitis media, the pharyngeotympanic tube acts as a conduit for pathogens in the nasopharynx to the middle ear. Inflammation causes buildup of fluid or pus. Tympanic membrane bulges painfully into the auditory canal. This interferes with hearing, and this is, again, acute otitis media. Healthy tympanic membrane viewed through that device, I'll think of the name of it in a minute. Tympanic membrane in acute otitis media, and then we have a tube inserted into the tympanic membrane so the fluids can drain, and this doesn't happen. Children are most susceptible to acute otitis media. Pharyngeotympanic tubes are much shorter and more horizontal than in adults. Also more frequent upper respiratory infections because the immune systems don't work yet. This is commonly treated with antibiotics, although some experts waiting to see if the infection will clear on its own. Those experts are not the ones that have the screaming 2-year-old living with them, because I can assure you the parents of the screaming 2-year-old want the antibiotics and want the infection cleared up as quickly as possible. Even with treatment, acute otitis media can progress to otitis media with effusion, which is where the infection has cleared but the fluid remains. This interferes with movement of the tympanic membrane for normal hearing. Persistent otitis media externa can be, infusion can be treated with laryngotomy, which is a small hole that is pierced in the membrane to drain the middle ear. A small tube can be inserted into the hole to prevent the infection from recurring, and the tympanic membrane heals and pushes the tube out within a year or two. Some children will have to have these replaced several times during childhood, just depending on the particular child. The inner ear is the cavity within the temporal bone, subdivided into 3 regions. You have the cochlea, the vestibule, and the semicircular canals. The bony and membranous labyrinth. Each labyrinth is a network of serpentine pathways composed of either bone or membrane, respectively. The bony labyrinth opens pathways that make up the inner ear chamber. The membranous labyrinth is membranes that line the bony labyrinth, containing the fluid called the endolymph. The endolymph is a fluid in which potassium ion concentration is greater than sodium ion concentration, similar to intracellular fluid. The perilymph is the fluid between the bony and membranous labyrinth. The sodium ion concentration is greater than potassium ion concentration, similar to that of the extracellular fluid. Both fluids play a role in transducing sound waves and head movements into electrical signals. The vestibule is located medially to the middle ear. The wall features the membranous oval window, houses the auricle or the utricle and the sacculae, both parts of the membranous labyrinth, the endolymph-filled chambers, and both contain receptor cells. The vestibule conveys stimuli about head tilting and linear movement via neurons of the vestibule division of the vestibular cochlear nerve, or cranial nerve number 8. The semicircular canals contain 3 tubes of the membranous labyrinth, sometimes referred to as the semicircular ducts. You have the anterior, the posterior, and the lateral semicircular ducts are oriented at right angles to one another. They can detect rotation movement of the head in any body plane. The ampulla is an enlarged bulb at the base of each duct, contains receptor cells innervated by the vestibular division of cranial nerve 8. Again, that's the vestibulocochlear nerve. And here is semicircular canals. There's the bulb, the ampulla on each end. Here's the temporal bone. Here comes in the vestibulocochlear nerve. Here's the vestibular nerve. Here's the cochlear nerve. And here is one nerve. That's why we refer to it as the vestibulocochlear nerve. It diverges with one tract going to the cochlea and another tract going to the vestibules of each of the semicircular canals. The cochlea features a membrane-covered opening called the round window, which separates the middle ear and the inner ear. The cochlear duct is a spiral-shaped segment of membranous labyrinth, which is connected to the sacculae, and it is filled with endolymph. The cochlear ducts twist around a screw-shaped bony core called the motulus, and then terminates at the tip of the cochlea. The scala vestibuli is a chamber within the cochlea. It is superior to the cochlear duct and is continuous with the vestibule near the oval window. The scala tympani is a chamber within the cochlea. It's inferior to the cochlear duct, and it terminates in the vestibule near the round window. Both of these chambers surround the cochlear duct between them and continuous with one another at the tip of the cochlea. The vestibular membrane is a sheet of epithelium that forms the boundary between the cochlear duct and the scala vestibuli. It separates the endolymph from the perilymph, and it is a barrier to maintain ion homeostasis of each of these fluids. The cochlear duct and the scala tympani are separated by the basilar membrane, which is composed primarily of collagenous connective tissue. It's connected to a small piece of bone that projects from the bony labyrinth. The cochlea basilar membrane becomes more flexible as it gets wider, which allows it to detect variations in sound pitch, supports the spiral organ within the cochlear duct. This structure contains receptor cells for hearing, also converts sound wave energy into electrical impulses, and it travels to the brain by axons in the cochlear division of cranial nerve 8. And here is a cross section of the inner ear. There's the oval window, the round window, and there's the cutaway view of the cochlea. Here's the cochlear nerve feeding into each one of these spiral ganglions. There's the scala tympani, the scala vestibule containing the perilymph. There's the scala media, and there's the basilar membranes. All right. Physiology of hearing. Sound must be understood to truly appreciate hearing. Ears detect and transduce sound waves into electrical signals that can be processed by the brain. Sound waves are generated by the displacement of air molecules in response to a moving object. The adjacent molecules collide with one another, thus transferring energy, and that transferred energy becomes a wave. That wave then radiates away from objects in 3 dimensions. It goes up, down, and out. Sound can be described in terms of pitch and loudness. Every sound has a certain pitch. That's determined by how many times an object vibrates back and forth during a certain period. Pitch is measured in what is called hertz. Human ears can detect sounds between 20 hertz and 20,000 hertz. That is our hearing range. Below 20 and above 20,000 are sounds we cannot hear. Loudness or amplitude is determined by the different areas of high and low pressure. Soundness is measured in decibels. So pitch is measured in hertz. Loudness or amplitude is in decibels. The greater force placed on an object causes greater differences between the waves of high and low pressure, producing louder sounds. The harder you hit a drum with the greater force, the louder the sound is going to be. And there is plucking a string makes it vibrate, transferring energy that radiates through the air molecules. Here's the amplitude, tells us the pitch, high amplitude, low amplitude. Frequency is pitch, loudness is amplitude, frequency is pitch. So high frequency, very short waves, low frequency, longer waves, low amplitude, high amplitude. And this doesn't show it all that well, but if you notice, while the peaks of these are very different, the length is very similar. You measure from here to here, you would see that it's the same as from here to here. Whereas this, when you're taking pitch, this is the low note, this is the high note. Sound transmission within the inner ear. The sound waves must be funneled from the cone-shaped auricle into the auditory canal where they strike the tympanic membrane. Structure and function, made the way it is so it can do its job, and it can do its job because of the way it's made. Sound waves then cause the membrane to vibrate at the same frequency as the sound. The vibration of the membrane is transferred to the ossicles in the middle ear, which transfer the vibration to the oval window, which leads into the middle ear. The vibration at the oval window then generates waves in the fluid in the inner ear. mostly the perilymph in the vestibule and the scala vestibuli. More force is needed to generate waves in fluid versus air. This is the function of the ossicles in the middle ear. The ossicle acts as a lever system, amplifies the vibration differences between the larger tympanic membrane and the smaller oval window to compensate for the transfer of sound waves from air to fluid. Pressure waves generated at the oval window are then transferred to the perilymph of the vestibule, then to the scala vestibuli of the cochlea. High frequency vibrations take a shortcut through the cochlear duct to reach the scala tympani, which causes the endolymph and the basal membrane to vibrate. Vibrations occur close to the base of the cochlea, where the basilar membrane is narrow and stiff. Low frequency vibrations or sound waves, however, have to travel further into the cochlea, where the basal membrane is wider and more flexible. Very low frequency sounds outside of our hearing range travel to the tip of the cochlea, where the scala vestibuli is connected to the scala tympani at the helicotrema opening. Sound waves then travel back to the vestibule along the scala tympani, causing the round window to vibrate. The round window is essential for allowing fluid to move within the inner ear. So again, sound waves come in, vibrate the tympanic membrane, which causes this lever system, the three bones, to oscillate, which then causes this membrane, and you have a high frequency sound which very quickly comes back, or the low frequency sound that has to go further along the nerve before the cochlear duct before it can head back. Pitch is determined by which area of the basilar membrane vibrates. Loudness determined by how much of the basilar membrane vibrates. The spinal organs rest on the basilar membrane, which allows for transfer of sound wave energy to the region where the vibrations can be converted into electrical signals. Those electrical signals are then sent to the brain for interpretation. Within the spiral organ, you have hair cells, which are hair-like. They look like hair. Receptor cells within the organ, they are grouped into inner and outer hair cells. The inner hair cells form a single row of cells. These are primarily responsible for detecting sound. The outer cells are in rows of 3. Each hair cell is equipped with a microvilli called a stereocilia, which extends from the cell into the endolymph of the cochlear duct. The stereocilia vary in length. They will be arranged from the longest to shortest, and they will flex at their junction with the hair cell body. And again, here are those stereocilia. Here are the cells, and you see they vary in length, and they will vibrate according to the sound coming in. The tectorial membrane is a stiff structure that extends over the hair cells. It sandwiches the cells into their stereocilia between the tectorial membranes and the basilar membrane. The stereocilia on outer cells are in contact with the tectoral membrane. Vibration of the basilar membrane causes these hair cells to move toward and away from the tectoral membrane. The tallest stereocilia will bend as they bump into the tectoral membrane, causing all the linked stereocilia to bend in the same direction. And you see here they all are arranged. And when the signal gets here, this one will bend, which will in turn cause all of those to bend in a like fashion. The basilar membrane will then move up toward the tectoral membrane, bending the stereocilia toward the tallest stereocilium. The bending stereocilia opens potassium ion channels, which depolarizes the hair cell. Then the potassium ion concentration in the endolymph is high enough to cause ions to flow into the depolarized hair cells instead of flowing outward to hyperpolarize the cell. Again, this is an example of that gradient core principle. The depolarized hair cells will then release their neurotransmitters, triggering action potentials in the axons of the cochlear division of cranial nerve 8. And again, there as it moves up, you see the bending, which releases the neurotransmitters, which then sends the action potential along that axon. The electrical impulses get transduced from sound waves, then they travel along axons of the nervous system along the following pathway. The auditory signals travel through the axons of cochlear portion of the vestibulocochlear nerve. The axons from the cochlear nuclei contact the superior olivary nucleus in the pons, which is important from information from both ears is compared for determination of location of the incoming sound. That auditory stimuli is then sent to the inferior colliculus of the midbrain, which is involved in the startle reflex that occurs in response to unexpected sounds. How many of you have been sitting somewhere and somebody's dropped a book and you jumped? The auditory stimuli are relayed to the medial geniculate nucleus of the thalamus. The thalamus then sends the signal to the auditory cortex in the superior portion of the temporal lobe. At the primary auditory cortex, that's where we become conscious of the incoming sound. We analyze it for location, pitch, and loudness. In the primary auditory cortex is connected with other regions of the temporal lobe, which are involved in language and the limbic system. And remember, the limbic system is our emotion and memory area. And again, here is the pathway. Here's the sound coming into the mid of the inner ear, signals coming in, connected nuclei of the pons, cross up into the stimuli being generated by the thalamus, and then out to the areas of the brain for the primary auditory cortex. Tinnitus, this is a word that I have pronounced tinnitus forever, but recently I've heard people starting to pronounce it tinnitus. I'm going to continue to say tinnitus. This is a sensation of hearing noises in the absence of any actual sound. The noise is usually described as a ringing or a buzzing, a whistling, or a hissing. It is not a disease process in itself. It is usually a symptom of some underlying problems. About 1 in 5 adults suffer from some form of tinnitus. The most common cause is hearing loss due to damaged stereocilia of the cochlear hair cells. Those cells are either bent or misshapen, or they are so depolarized enough to release neurotransmitters onto the cochlear nerve endings, even though there is no stimuli to release those neurotransmitters. Also could be a result of fusion of the middle ear ossicles, inflammation or irritation of the tympanic membrane. Some medications can cause tinnitus, or damage of neural pathways in the central nervous system. In many individuals, no cause is ever identified. In some cases, the tinnitus is temporary. For example, large doses of aspirin can cause it. For many people, it is permanent. I have a friend who has had a problem with ringing and buzzing in his ears for the past several years. Nothing seems to help. Antidepressant medications can help in some cases. Many people find relief by using a fan or some other source of white noise to block sounds, especially while sleeping. My friend, that's one of the things he does. You will never go to his house and there's not a radio or a TV or a fan or something going in the house. Excuse me, just to give him that little bit of white noise and that sort of block out the sounds of the buzzing in his ears, because there is something for him to actually be hearing. Hearing loss is defined as deafness. Deafness is defined as any type of hearing loss. Can be classified into 2 very broad categories: conduction and sensory neural. Conduction hearing loss is due to a problem in the outer or middle ear, which prevents the sound from reaching the inner ear. Could be something as simple as an excessive wax buildup, a middle ear infection, a perforated eardrum, or fusion of the auditory ossicles. And here, in this case, vibration is reaching the inner ear through the temporal bone, but it's with much less accuracy than conduction through air. Many forms can be corrected. If all you have is a buildup of earwax, remove the excess earwax, and your hearing should come back. Repair the perforated tympanic membrane if you have a perforated membrane. Sensorineural hearing loss, however, is a deficit in the cochlea or other part of the neural pathway. Two different types here as well. You have sensory hearing loss, which is where the action potentials cannot be generated in the cochlea, usually due to hair cell dysfunction. Exposure to loud sounds or certain medications may damage hair cells. Also, you can lose stereocilia as we age, especially those in the proximal part of the cochlea. This is responsible for detecting high-pitched sounds. So those of us who spend our teen years listening to a lot of really loud music may have some form of sensory hearing loss. Hearing aids can help mild to moderate cases of sensory hearing loss. Cochlear implants may also be an option for this type as well. Neural hearing loss is where the signals fail to travel through the cochlear branch of the vestibulocochlear nerve or the central nervous system pathways. Typical causes of this include strokes or tumors, such as an acoustic neuroma, which is a benign tumor outside the brainstem at the junction of the medulla and the pons. Cochlear implants are not effective for neural hearing loss. As far as I know, there are no effective treatments for neural hearing loss. Cochlear implants is a surgically implanted electrode that bypasses the damaged hair cells and directly stimulates the cochlear nerves. They do not fully restore hearing. They enable a person to perceive enough sound to understand speech or hear warnings such as smoke alarms. But the implant consists of an external microphone, there's my biology training coming out, usually worn just behind the ear, sends sound waves to a processor which converts the waves into electrical signals. It will also include an external transmitter which sends signals to the internal receiver, which is implanted beneath the skin. The receiver then relays the signals to the long, thin bundle of electrodes threaded through the cochlea, which stimulates the nerve endings of the vestibulocochlear nerve. Success of an implant varies. Children who are born deaf or suffer hearing loss shortly after birth usually get implants before age 3. The period of maximal language and speech acquisition often learn to speak normally. If a deaf child received the implant after age 9, it is unlikely that normal speech would develop. Children and adults who lose their hearing after acquiring speech and language often do really well with these. No longer have to solely use sign language or read lips. The radio entertainer Rush Limbaugh had, I don't remember what ear issue he had, but he had an ear issue where he virtually went deaf over just a few weeks. You know, he started to notice his hearing loss and he lost his hearing. But he had a cochlear implant, and for the last several years of his life, he was able to function hearing using the cochlear implant. Regardless of age, extensive training is required to use the implant successfully because you have to learn, you basically have to relearn how to hear, because you have to learn how to block out everything that's coming around you and all of that what we would refer to as noise, and concentrate solely on what you're trying to listen to. So there's the big picture of hearing. Sound waves come in, vibrate the tympanic membrane, which in turn malleus, incus, and stapes begin to move the oval window. The signals come in, then they enter the nerves and the vestibulocochlear nerve, go through the I'm gonna think of the word I'm looking for here in a minute, the vestibule of the cochlea, and they go to the brain where they are processed. And we will pick up with the physiology of vestibular sensation, or our sense of balance, a little bit later.